My name is Oliver Renner. I'm heading the communications function of Bayer's pharmaceutical division. Headquarters of the pharmaceutical division are here in Berlin. And you might ask yourself why we are hosting the American Academy here today at our facility. It's because our company is supporting the Academy a long time, for decades, I would say. It's the predecessor company, Schering AG, already started with this because the companies always believed in the transatlantic dialogue and its importance. And as you all know, or might know, or will know, the Academy is an important element in order to nourish these discussions and the transatlantic exchange through their fellowship program, which is a core element of the Academy's being. We have Daniel Benjamin, who is the president of the Academy here tonight with us, and of course, Bayer Fellows for Biotech and Health, Harvey Feinberg, and his wife, Mary Wilson. And I'm very honored that Mary will speak to us tonight because it's basically a very special situation we have here. Normally we sponsor only one fellowship a year, but we got two fellows this year, and it's a great honor to have you here, Mary. And with this, I hand over to Dan.
I think I'm mic'd.
You're mic'd.
I'm mic'd, yes. Thank you. Well, it's a great pleasure to be here. I am, as was advertised, Daniel Benjamin, and I'm the president of the American Academy in Berlin, and I'm really pleased to be in this room. I have not been in a lecture room like this since I left the university I was working at 3 years ago, and I love the feeling of being back. Anyway, I want to welcome you to the Bayer Lecture in Health and Biotech, Confronting the Threat of Antimicrobial Resistance, which will be delivered by Mary E. Wilson. Mary has a title that is staggeringly long. She is the clinical professor of epidemiology and biostatistics at the University of California in San Francisco, as well as adjunct professor of global health and population at the Harvard T.H. Chan School of Public Health. Now, I can tell you, knowing a bit about my own institution, that when its founders convened and decided that they would create the Academy, the word antimicrobial was not one that they were thinking about having in any lectures in the near term. But I am absolutely thrilled. By the way, among those people were Richard von Weizsäcker and Henry Kissinger. Kissinger, I think now these days probably would use the word antimicrobial since he's into artificial intelligence and things like that. But suffice it to say, in the mid-90s, this was not on their mind. But I am thrilled that we are having this talk. I think we've all learned, even us non-scientists in the last few years, just how important public health is to the functioning and wellness of our society. And so I'm really delighted that we are doing this here, and I particularly want to thank Bayer AG for its support for this fellowship, which I think has, in fact, helped point the Academy in the direction of a real important expansion of its activities. And I want to thank my friend Oliver Renner, who is here, of course, and Stefan Ulrich, who has been such a champion of the relationship between Bayer and the Academy. Bayer has been a supporter of the Academy for almost 20 years, and I couldn't be more grateful. It's been a terrific partnership. And of course, I should add that we do not take public funding, so the support of firms like Bayer is absolutely essential for us. We also plan to continue this as Oliver mentioned. The fellowship is meant to bring one brilliant mind a year to the Academy. I'm really delighted that in only our second year we managed to bring two in the form of Mary Wilson and her husband, Harvey Feinberg, and I certainly hope that in future years we can try for 3 or 4, Oliver, so maybe we should discuss that later. Now, to introduce our speaker, Mary, as I mentioned, has that very long title, dealing with epidemiology and biostatistics. Her interests include the ecology of infections, the emergence of microbial threats, travel medicine, tuberculosis, and vaccines. She has a list of publications significantly longer than my arm. She has contributed greatly to our understanding of how bacteria develop resistance to antibiotics and the mechanisms that underlie this phenomenon. She is widely regarded as one of the foremost experts in this field, and we're really honored to have her with us today. She has an MD from the University of Wisconsin. Go Badgers, is that right? Okay. And completed an internal medicine residency and infectious disease fellowship at the Beth Israel Hospital in Boston. She was the chief of infectious diseases at Mount Auburn Hospital, one of Harvard's affiliated community teaching hospitals in Cambridge, for more than 20 years. She is a fellow in the Infectious Diseases Society of America, the American College of Physicians, and the American Society of Tropical Medicine and Hygiene. She has served on a whole range of different national blue ribbon advisory committees and 5 committees alone for the National Academies of Science, Engineering, and Medicine. She has worked in Haiti at the Albert Schweitzer Hospital and led the Harvard Brazil Collaborative course on infectious diseases taught in Brazil. Among the things that we really need to feel sorry about for Mary, she was a resident scholar at Bellagio in 1996, and she was also a fellow at the Center for the Advanced Study in Behavioral Sciences at Stanford. In addition to all of her many publications and other distinctions, there is one other really important intellectual accomplishment Mary has that seldom shows up in print, and that is, as we have seen at the Academy, she plays the role that might be analogized as autocorrect for her husband. More than once we have seen him in conversation, and she has done what is known as, in English as pursing her lips when he was, shall we say, too far forward over his skis. That's a government phrase to talk about getting ahead of oneself. It has been a complete delight to have them both at the Academy, and really just an absolute gift for us. So before I go on too long and start gushing, I'm going to pass the microphone over to Mary and hear what she has about antimicrobial resistance, origin, scope, and solutions.
Well, thank you, Daniel, and thank you to the American Academy in Berlin and to Bayer for supporting the fellowship that Harvey and I have, and also for hosting this event today. Antibiotics have been available since the 1940s and have saved millions of lives. But antimicrobial resistance now threatens global health. Resistance has been called a silent tsunami, a silent pandemic. And today what I would like to do is provide, through 10 observations, I'm going to talk about the natural and the anthropogenic origins of antimicrobial resistance. I'm going to describe some of the sources and scope of antimicrobial resistance. I will lay out a framework for thinking about solutions to antimicrobial resistance, which cuts across multiple disciplines, geographies, species, and sectors. And finally, throughout all of this, I will emphasize the importance of a One Health perspective, the need to take into account humans, animals, and our shared environment in approaching the problem of antimicrobial resistance. So, observation number 1, antibiotics are ancient and arise in nature. Bacteria live in communities. They compete for resources. They communicate with each other. And in fact, they produce substances that can kill, inhibit, or even promote the growth of other microbes. In 1928, the scientist Alexander Fleming observed on an agar plate in the laboratory that there was a clear zone surrounding a colony of a fungus that actually turned out to be Penicillium notatum. He surmised that this fungus must be producing a substance that was inhibiting bacteria. And over the next many years, in fact, more than a decade, he, with the help of other scientists, actually extracted, purified, and then made in sufficient quantity that they could use this substance, which was penicillin. But they first treated a patient with this in 1941. Another scientist, Salman Waxman, coined the term antibiotic, by which he meant substances produced by or derived from other living organisms with the capacity to destroy or stop the growth of bacteria. He spent his life working on soil bacteria, and he actually discovered that one of them, Streptomyces griseum, produced a substance that he called streptomycin that not only inhibited a number of pathogenic bacteria, but also was active against the bacteria that caused tuberculosis, Mycobacterium tuberculosis. So bacteria produce the substances that originally were the original antibiotics and have served as a blueprint for making other antibiotics. Now, some of the antibiotics, like ciprofloxacin, are completely synthetic, but bacteria provided the blueprint for these. The term antimicrobials is broader than antibiotics, but we often use the two terms antibiotics and antimicrobials interchangeably. But by antimicrobials, we also include drugs that are active against viruses, against fungi, and against parasites like the malaria parasite. So key message number 2, antibiotic resistance is ancient. We said that bacteria produce antibiotics, but bacteria and other microbes also produce substances that thwart or that counter the effect of antibiotics, and they have been doing this for eons. People have identified ancient bacteria that have had no contact with humans or with human-derived drugs, and have found that antibiotic resistance genes were present in bacteria going back not only thousands, but millions of years. Antibiotic resistance is defined as the capacity of bacteria to survive in the presence of antibiotics. It's a biological trait of bacteria. The antibiotic does not weaken. The bacteria change in ways that enable it to survive despite that. And resistance elements are found in bacteria that produce disease and those that do not. And in fact, resistance and the capacity to produce disease are different attributes of bacteria or other microbes. Bacteria have the capacity to replicate rapidly every 20 to 30 minutes, which gives them enormous capacity to change rapidly. So antibiotics or bacteria have a huge number of ways that they can resist the effect of antibiotics that have developed, again, over eons. They can block an antibiotic, or they can block the antibiotic from actually entering the bacterial cell. A really common mechanism is to produce an enzyme called beta-lactamase that actually cleaves the beta-lactam ring that is essential for the function of penicillin and many other antibiotics. Some bacteria can even pump an antibiotic out of the bacterial cell after it enters. bacteria have a huge library of resistance mechanisms that they can draw upon to resist the effect of antibiotics. Key message number 3, antimicrobial resistance is a major killer today. The best study that gives recent estimates of the burden of antimicrobial resistance globally was done by the Institute for Health Metrics and Evaluation in Seattle. And actually, the Robert Koch Institute here provided some of the data for this massive study that was published last year. And what they found was that globally there are about 5 million deaths annually associated with antimicrobial resistance, and about 1.25 million deaths directly attributed to antimicrobial or bacterial antimicrobial resistance. It's notable that of the top 6 pathogens causing deaths globally from resistance, there's a vaccine available for only one of these, Streptococcus pneumoniae. And this is a slide that's borrowed from the researchers at the Robert Koch Institute that they had developed, but it shows that antimicrobial resistance is in the same league with other major killers globally. It is shown in red. The dark red are those directly caused by antimicrobial resistance, and the lighter red are those associated with antimicrobial resistance. Today, antimicrobial resistance causes more deaths than HIV/AIDS or malaria. And this is just bacterial antimicrobial resistance. Resistance is also a problem with viruses, with fungi, and with parasites like malaria. But this particular study looked at only bacterial resistance, and that is what I'm going to be focusing on in my comments today. So the burden of antimicrobial resistance is higher in low and middle income countries, and particularly in sub-Saharan Africa and in Southern Asia. But in fact, the same group that did the study I just mentioned also looked specifically at resistance in the WHO European region, and estimated there were more than 500,000 deaths annually from bacterial antimicrobial resistance, and more than 100,000 deaths. This is on an annual basis. Key message number 4, antimicrobial use drives resistance. Antibiotics kill sensitive bacteria in a population. Those that are resistant survive, proliferate, and become predominant. When penicillin was first introduced in 1941, within about 6 years, about a quarter of the staphylococci in hospitals where they were using the penicillin already had become resistant to penicillin. When I did my infectious disease training in a Boston teaching hospital, about 90% of the bacteria, of the staphylococci, were resistant to penicillin. During the second year of my fellowship, I spent part of the year working in Haiti, in rural Haiti, in Deschapelles, at the Albert Schweitzer Hospital, and there about 90% of the staff were still sensitive to penicillin. They simply had had very little antibiotic exposure over the years. And we see with antibiotic after antibiotic that over time, bacteria increasingly become resistant. And this particular figure shows a particular type of antimicrobial resistance, the extended spectrum beta-lactamase, over a period of 25 years. And it looks at 66 studies that were done globally. And what you see is a progressive increase in the presence of this particular resistance gene over time. And this is in people who were carrying it in their gut bacteria. At a country level, one can show a significant association between the per capita use of antibiotics and resistance genes carried in gut bacteria in healthy individuals who are not taking antibiotics. And this is a study that was just published. You may notice that the Netherlands and Germany both are on the end with low use of antibiotics and low level of antibiotic resistance genes. Key message number 5. Most antimicrobial use globally is in food animals. Antibiotics are not just for humans. They're used in cats, dogs, goldfish, honeybees, but they are also used extensively in food animals, particularly pigs, chickens, and cattle. In fact, almost 3/4 of antimicrobial use globally is used in food animals. Why are they used so extensively? Well, they are used to treat and to prevent infections, but much of the use in food animals is for what is called growth promotion. When antibiotics were first developed, as far back as the 1950s, people observed that giving animals low doses of antibiotics made them gain weight faster. Antibiotics were cheap, and particularly under conditions of high density and poor sanitation, it made economic sense to give animals antibiotics. And in the most recent study published by the Food and Agriculture Organization, FAO, looking globally at 190 countries, they found more than 1/4 of them still used antibiotics for growth promotion. The EU passed regulations a long time ago prohibiting the use of antibiotics for growth promotion, and other countries have passed regulations more recently, but a number of countries continue to do this, continue to use them for this today. Well, you might think that animals would have their own antibiotics, but in fact, what you see is that the same classes of antibiotics are used in humans and in food animals and in aqua farming, and to some extent even in plants and honeybees. Antibiotics generally have been developed for use in humans and then are subsequently used in animals. And there's been a movement to try to restrict the use of antibiotics that are critically important for human health from use in animals. And there's been some success in this area. In the United States, honeybees, interestingly, are regulated as food animals because they produce honey and pollen. And even though their most valued, their greatest value is for pollination, but only a limited number of antibiotics are used in are regulated for use in honeybees in the United States. This simply shows the tonnage of antibiotic use by area in 41 different countries around the world. This is only veterinary use in food animals, and what one sees that overwhelmingly the greatest amount, in fact about 40%, 45% of global veterinary use is used in China, and this is largely for growth promotion. This gives a snapshot of all antimicrobial use consumption globally over more than a decade. And what I find really striking is that, in fact, pigs receive the greatest number of tons. They are shown in pink. So the pigs are in pink, the chickens are in sort of an orange, and humans are at the bottom on the gray bar. So globally, many more antibiotics, as I said, are going into food animals. The amount going into use for aquaculture is much more uncertain than it is in the food animals. This helps provide context by giving the biomass of the various species. And if one had a giant scale and you put all humans on it, and then you subsequently put all food animals on it, and then you weighed the wildlife, what you would find is that food animals have about 3 times the biomass of humans today. In fact, there's been a rapid increase in the production of food animals as living conditions as economics have improved in many low and middle income countries. There's been a desire for more animal protein. So there's been a major increase in Asia and in Africa for animal protein. This shows the fish, and the pink shows what the caught fish. And what one sees is that globally there's been a rapid increase in aquaculture, so that today about half of all seafood is produced by aquaculture. And in terms of antibiotic use in aquaculture, more than 90% of this is in the Asia Pacific. Key message number 6, antimicrobial resistance moves. Bacteria transfer resistance from one bacteria to another. They pass it vertically from bacteria to progeny, but they also pass it laterally or horizontally. And bacteria can even pass resistance genes to unrelated bacteria. Not necessarily just E. coli to E. coli, but they can transfer resistance to other species of bacteria. And they have multiple mechanisms for doing it, but sometimes they use mobile genetic elements and can sometimes even transfer resistance to multiple antibiotics at the same time. People also transfer resistant bacteria from one person to another, from mother to child, within households, within hospitals. But there's also long-distance transfer or dispersal of resistant bacteria and resistance genes. This is largely through travel and trade. Food is a really important mechanism for the transfer of resistance from animals to humans, but also globally. We have a very long food chain, and it is one of the mechanisms by which resistance is transferred. Wildlife, even if they've never been given antibiotics, can often pick up resistant bacteria. They feed on water pools that are contaminated or on waste that contains resistance genes. A number of specific mechanisms of resistance have been tracked globally, and one that was particularly prominent was what was called the New Delhi metallo-beta-lactamase that got a lot of publicity because some of the early reports came out of New Delhi, even though that's not where the original isolates occurred. But these particular resistance genes have made it possible to track the movement of some of these types of resistance around the world. And again, travelers have been really important in carrying resistance genes and allowing them to be introduced into new areas. The background rates of resistance vary enormously from one geographic area to another. And this shows the background prevalence of, again, this extended spectrum beta-lactamase around the world. And the size of the circle is indicative of the percentage of healthy people who carry that particular form of resistance. And you can see it very low in Europe and in North America, but very high in parts of Asia. And multiple studies now have looked at travelers who are going, say, from the Netherlands, traveling all around the world, and then looking at them after they return. And what you find is that it is very common for people, for travelers, to acquire resistant bacteria even if they have no symptoms whatsoever. And what they acquire reflects the background rate of resistance in the places that they have visited. And in some instances, some of the travelers to parts of Asia, half or more may carry resistant bacteria when they return. Key message number 7. Antimicrobial residues and resistance genes widely contaminate the environment. Antibiotics have an afterlife. If an antibiotic is given to a person or a pig, it does not disappear. If a person takes a dose of a common antibiotic, amoxicillin, for example, within 6 hours, about 60% of that comes out in the urine, unchanged, and still biologically active. In high-income countries, in Germany and many parts of the world, the waste is flushed and ends up going to a modern water treatment facility. But much waste, much human waste globally goes into soil, into water, into the environment. And certainly most of the waste from animals globally is not treated before it enters the environment. Modern treatment plants are designed to help make water safe to drink, but in fact, water treatment plants do not eliminate all residues of antibiotics, although they markedly reduce the amount. So an effluent from a wastewater treatment plant will still have residues of antibiotics in it when it goes into whatever water source that where it goes. And I'd like to show 2 studies that give some sense of the widespread pollution we are now seeing with antibiotics and resistance genes really globally. There's a study from China that looked at a study where scientists took samples from 18 estuaries along the entire eastern seaboard, over 4,000 kilometers of coastal China, took the samples back to the laboratory and looked for the presence of antibiotic residues and resistance genes, and found that they were present in them, and also found that there was a correlation between the amount of antibiotics, and they found all of the common antibiotics, and correlation with resistance genes. Keep in mind, these are waters that are being used for recreation, for irrigation, for use growing marine food that that becomes part of people's diet. In the Netherlands, soils have been archived as part of the agricultural research program. And a few years ago, researchers went back, pulled samples going back to 1940, before antibiotics were used, and then looking at many other samples through 2008. And what they were able to show is that there was a progressive increase over the decades in resistance to the commonly used antibiotics, so beta-lactams, to tetracyclines, and to erythromycin. So Key message number 8, antimicrobials cause collateral damage. When many of us grew up, we were taught bacteria are bad, antibiotics are good. But in some sense, we have that partially backwards. I think we are coming so much more to recognize that, in fact, our microbiota, all of the bacteria, the assemblage of bacteria and other microbes that live in and on us, in fact, do essential functions for life. And in fact, when a person takes an antibiotic for a strep throat, the antibiotic diffuses to tissues throughout the body. And it may kill the strep in the throat, but it also will kill bacteria any place in the body that it reaches that are sensitive to it. And if it's a broad spectrum antibiotic, it will kill even a wider swath of bacteria. So there's a lot of off-target killing of bacteria when one takes an antibiotic. The microbiota, again, or the assemblage that we have in and on our bodies, performs essential service. It's passed from mother to infant, or that's the beginning of it, and then is modified by diet and other activities. But we now know that the microbiota has an influence on nutrition, metabolism, immune response. It also does something called colonization resistance. A healthy, heterogeneous microbiota can help resist the invasion of bacteria that can cause disease. There's also increasing awareness, increasing studies showing that the microbiota and a disturbed microbiota may play a role in some of the chronic diseases like obesity, diabetes, allergy, and autoimmune diseases. Key message number 9. Modern healthcare relies on safe and effective antimicrobials. Healthcare as we know it today developed during an era when safe and effective antibiotics were available. Childbirth, surgery, organ transplantation, all of these and many more depend on the presence of effective antibiotics. This was a study where a group tried to model the consequences if some of the bacteria became resistant, if some of the bacteria became resistant or if all of them did, but looking at the increase in mortality from these common procedures if there was an increase in antimicrobial resistance. And in fact, it would really change the risk-benefit for many medical procedures if, in fact, the mortality was increased because of antimicrobial resistance. Finally, making more antimicrobials alone will not solve the problem of antimicrobial resistance. We have seen repeatedly, we've lived through really a golden era when over many decades we've had, I would say the golden era is over, but we had many decades where there was one antibiotic after another that we could turn to when bacteria become resistant. But I think we're now bumping up, we're reaching a place where in fact it is more difficult to keep creating new drugs quickly that are active against the resistant bacteria. And I think we need to look to other solutions. And I find it useful to think in two major categories. One is what we can do, one of the things we can do right now, and that is reducing the use of the antibiotics to try to preserve what we currently have in terms of the antimicrobials, we could go a long way with better use of what we currently have. I don't know what the situation is in Germany, but I know in the United States it's been estimated that as many as 30 to 40% of the courses of antibiotics are given for diseases or symptoms that do not benefit from the use of antibiotics. If we had better diagnostics, we could do a much better job of using antibiotics or the right antibiotic for a particular infection. If we immediately knew the sensitivity of a bacterial pathogen so that we could use the right spectrum and not have to use a broad spectrum antibiotic, it would be beneficial. We should eliminate the use of antibiotics for growth promotion in animals. Many countries have done this. It requires investment to do this, and this has been possible even in aquaculture in some of the Scandinavian countries, where they've been able, through vaccinating fish, if you can believe it, to reduce use of antibiotics in aquaculture. But again, it requires attention to living condition, the conditions for the growth of the animals to do this. We could do much more with education, not only of medical personnel, but there are many people who expect to receive an antibiotic if they have a cold or a cough. And many people still don't understand that antibiotics do not work in that setting. The other major category is to reduce the need for antibiotics, and we could eliminate a huge need for antibiotics if we did this. We could start with clean water and sanitation everywhere, better infection control everywhere. Healthcare-associated infections will occur, but one can do a better job of preventing in hospitals and from facility to facility. There's huge benefit to be gained from vaccination. And this is not, we would start with using the vaccines that we already have, but not just vaccination for bacterial infections like Streptococcus pneumoniae, but in fact, giving vaccination for viral infections can reduce antibiotic use. For example, with influenza, there have been studies that have shown that vaccinating for influenza can reduce antibiotic use in a population. Not only do people have fewer febrile illnesses, but you can reduce the secondary bacterial infections. Using rotavirus to prevent diarrhea in children, and upcoming the respiratory syncytial virus. So we also need to develop new vaccines, but we could do a lot in preventing the need for antibiotics by improved use and development of vaccines. And although most vector-borne infections we do not treat, do not benefit from antibiotics, infections like malaria and dengue, many patients with those infections end up receiving antibiotics because they have fever and they are sick and they end up in a hospital. So there's an enormous amount headway we could make by reducing the need for antibiotics. And finally, we need to develop new approaches to treatment. This includes antibiotics and other chemicals and substances that can actually treat or can kill bacteria. But there's really very active research going on in a number of other areas. Some of these include, rather than killing the bacteria, you disarm them. You intervene by avoiding the consequences of the toxins produced by the infection. We give antibodies to enhance the immune response of the host. And another one where I think we're going to see more and more work is using, in a sense, good bacteria to either replace or to outcompete the pathogenic bacteria. And we're already starting to see this in treatment of Clostridium difficile, the very destructive and unfortunately common infection, Clostridium difficile, that often occurs in hospitalized patients who have received a lot of antibiotics. And there are already approaches that are being developed that, again, using in a sense, good bacteria to replace the pathogenic ones. Use of bacteriophages, these are viruses, and one can find ones that are very strain specific and can really provide precision killing of only the bacteria that are causing the disease. And this is very labor intensive, and it's not clear this is going to have wide use, but it is something that can be used, is being used and is being looked at in a more systematic way. And finally, we do need new antibiotics. Those have been problematic, and we can talk about this more in the discussion, but antibiotics do not make good sense. Most pharmaceutical companies have gotten out of making antibiotics because antibiotics are used, and the more they use, the less effective they become. So it's been difficult to make a value proposition for the development of antibiotics. So these are the key messages that I have gone through. Let me just end by saying the prerequisites for any of these solutions is that they must be global. It will not work for a community, for a country, to a region, for a region to develop approaches to avoid antibiotic resistance, because we are connected in the biosphere. We must have a One Health perspective in dealing with antimicrobial resistance, and the surveillance, the systems, the interventions must deal not only with humans, but with animals and with the environment. And finally, the solutions must be equitable. I've mentioned overuse, which is common in many areas, particularly in some high-income areas. But at the same time, we have excessive use of antibiotics. We also have lack of access in many areas where people have either no access or no access to some of the latest and most effective drugs. So we really need to deal with those as well. But let me also leave you with a challenge, and that is, I think the term antimicrobial resistance, or AMR, has not really gotten people's attention. I don't think people understand the seriousness of the problem. Do we need a new name, a new way? It's not, it's not like Ebola or plague or some of the terms that people immediately understand in terms of the seriousness, but maybe we need to brand this in a different way to get more buy-in and attention. And with that, I thank you for your attention. I look forward to comments or questions.
I bring very little expertise to this, but I can claim honestly that I have laid awake at night worrying about antimicrobial resistance for many years. And I mean, one question I would ask, which you touched on at the end, is it would seem like it would be easy to make many more generations of antimicrobials, antibiotics, and yet I'm told it's not so easy. What seems to be the problem? Is it a market problem or is it a biological problem?
Both.
Both.
Certainly it's a market problem, because if you make an antibiotic, you want to sell it. You want to make as much as you can. You want to put it out there. people take antibiotics typically for a short period of time. It's not like a blood pressure or a heart or diabetes medication that you take for a lifetime. You take a short course, and that is it. The more you use an antibiotic, the more rapidly resistance to it develops. So the more you put it out there, the more you shorten its life. So antibiotics, in terms of a big market, have, I mean, it varies with the particular antibiotic, but scientists are really smart, but they have not yet found a way to develop drugs that can totally prevent the development of resistance. So, but it's partially market, but it's also biological. I mean, the bacteria have been at this for eons. They find ways to really get around pretty much anything we can throw at them. So it's really a dual problem.
And one more question, then we'll open it up to people who know something about the subject. What would a coordinated effort to fight AMR look like? Would it just be the profusion of best practices among doctors and people who raise livestock and things like that? Or is there even a possibility for coordinated governmental action?
Well, it requires involvement of so many sectors. And in fact, there is something called the Global Leaders Group, really trying at the highest levels to start bringing in agriculture, pharmacy, healthcare, education, I mean, really all of the sectors, the environment. I mean, you really need a coordinated and multi-sectoral approach. And there's something for everyone, including, as I said, the general public, because part of the use of antibiotics comes from interest generated from people. It's interesting, in the United States, there's a company some of you may have heard of, Purdue, that makes chickens. And
not Purdue Pharma.
No,
make other stuff.
These are the chicken. Yeah. These are the chicken Purdue people, but I think they make something like 500 million chickens a year. I mean, they produce a lot of chickens, but they decided, because of consumer pressure several years ago, they would make their chickens antibiotic free, starting with the egg, the chick, through the entire process. And it required investment, time, teams, and at each stage they used a lot more vaccines. They had to provide better living conditions, more space, but but it was consumer demand. And I think if consumers can be part of it, but again, people also need to know that again, people, I think, generally have the sense antibiotics are good. What harm can it do if I take a few pills of an antibiotic, and maybe it will be better for me? And I think we need to change people's thinking about the risk-benefit for those as well.
Okay, so let's open it up to the scholars and scientists among us. Please raise your hand, or I'll have to keep asking questions, and you'll be much less happy about that. We have a question right here.
Mary, I agree with you that we have to think global. But which efforts are being made that this problem really is attacked and try to cope with it globally? I mean, it's so complex. You mentioned that. It's farmers, it's fishermen, it's patients, it's doctors, and it's all kinds of populations all over the world. And it's low middle income countries, so there's no diagnostics. And how do you want to cope with it? What is the organization of coping globally with this problem? I can tell, for those who don't know, I was looking around. Konrad Reinhardt is not here. I see He is the founder of a Sepsis Alliance in Germany. And he is actually a retired professor because nobody else, I'm not saying that nobody else want to do it, but actively, this problem is not taking up. Of course, people are fighting sepsis, but there's no structure in this country except for the Sepsis Alliance, which is a one-man show in a way. Of course, he gets support more and more. There was a meeting just last week, but not sufficient support even in this country, and you mentioned that in other countries. So what are the efforts, what is needed to cope with this big problem?
Well, in fact, much is being done. I think it hasn't been sufficient. And I think part of what we need is greater general awareness from the general public. But there's been a lot of work around hospitals, nursing homes, dental offices, in agriculture. I mean, there's a very long list of what potentially can be done, but we need to do all of them simultaneously. One can't point to 2 or 3 ways that would solve the problem. I think the animal use is an important part of it, and I think generally the consideration of the environmental contamination with them has been under and unrecognized. One approach, and there's now a global consortium that's looking at some of the specific ways, is using wastewater to get better mapping of antimicrobial resistance. And I think COVID, one of the spin-offs from that, has been the increased attention to the use of wastewater for testing. I mean, there they were testing for the presence of virus, but it turns out there are a lot of things that you can test with wastewater, and that is available everywhere, and that may give better mapping of where it exists. But we really need interventions at all levels, and starting with school children in teaching them about some of the basic biology of resistance.
Questions? Paul? While you're there, we'll get to Paul second.
Oh. So to Paul next, or?
Paul will be after this gentleman, because he looks like he is a biologist.
Yes.
Paul is a Russian historian. So
I would just like to take the chance to add a couple more points to what Professor Gunton was raising. So I think there are definitely gaps which could be taken by non-governmental organizations, and that this has been well done by the Sepsis Alliance, I would agree. But on the other hand, there is also a bit of a blind eye among, in particular, I feel faculty of universities, to see what public health and public organizations are doing and have been doing for a long time. So, for instance, we do have in the local public health service, the regional and the national public health service, surveillance systems covering sepsis, antimicrobial resistance, and feeding those data back towards the clinicians and towards ECDC, WHO, and other global systems, and into those important burden estimations. Then beyond that, we support WHO and the Tripartite Office in going into countries, for instance, with the IHR mandate and the joint external evaluations. So a participatory process where we go and evaluate among all sectors with experts from FAO and from WHO and RKI and other institutions. We jointly go there and see what is there, what has been done against antimicrobial resistance. So I think strengthening that what has been already built up by public health sectors would also be a way to improve what we can do against antimicrobial resistance, instead of saying that there was nothing there. Because I've here strongly would advocate for there is a lot there already on the public side, but it is not well recognized often, and it does not have a comparable power to other institutions or companies that are out there that are trying to pick something of the attention on antimicrobial resistance. And that's what the Global Leaders Group is also advocating for, strong public health institutions that may assist and help to govern this complex problem.
Thank you very much. Paul.
In some ways, actually, the comments just there partly answered the question that I was going to ask. I mean, I guess I'm trying to understand. I mean, I'm impressed by the fact that the chicken producer Purdue is responding in the way that it has. It suggests that there's a level of consciousness that's already there, and some other initiatives have just been delineated. So I guess maybe my broad question is, and this goes to the question, the challenge you issued at the end too, like what is the language to use to describe this? Do we talk about antimicrobial existential catastrophe, or like what do we talk about? What is the place that it occupies in some of the most important institutions, the World Health Organization, this issue, I mean, the World Health Organization, in medical schools? I think maybe I don't fully quite see where you would say, I think it doesn't occupy a visible enough place or a prominent enough place because it's a serious problem, as you're saying. But maybe sketching out what do students in medical school, what do they learn about this particular problem? What are the initiatives of large organizations like governments, but above all, I suppose, global organizations for exactly the reasons that you've indicated? Maybe sketching that out might be useful for understanding the place that it occupies in the hierarchy of concerns that these organizations are facing. Thank you.
Well, thank you. Thank you, Paul. I mean, the World Health Organization, for example, a couple of years ago listed antimicrobial resistance as one of the top 10 global health threats to the world. And WHO has done a lot. And there have been many, and CDC in the United States, the top infectious disease society, the Infectious Diseases Society of America, has for many years been very involved in really trying to promote awareness. There's a lot going on by individual groups. I'm still not convinced that the general public, that there's general awareness of this as a problem, but there's an enormous amount going on. I think that there are many links between those in the agriculture side, but I think there's been enormous resistance in the United States to, and perhaps in some other countries, to limiting the use of antimicrobials. I mean, there's been interest. I mean, for example, in human health in the United States, you have to write a prescription if you want to use an antibiotic, and I'm sure that's true in Germany and in many of the high-income countries, but in many parts of the world, you can walk into any pharmacy, or even you can find in the airport any antibiotic you can imagine that you can buy. You can go into a feed store, an agriculture feed store, and buy a bag of antibiotics. And for food animals, typically the antibiotic is put either into the feed or into the water, and so it also can get dispersed into the environment. So there's an enormous, I mean, I perhaps understated the amount that is being done, but it's going to require effort across the board from many, many sectors, and coordinated activity as well. And we still have a lot to do.
I have a question regarding the topic of bacteriophages, because you said that this is something that is used these days rarely, but can be used as an alternative to antibiotics. It sounds like it's not very common and it's rarely used. Do you have a take on the future of that? Would that something, or would that be possible to use in the future instead of antibiotics? Because it sounded like it's rare and it's new, and if I'm informed correctly, at least in Germany, it's like it's not personalized medicine, but like individual medicine. So for each patient, it has to be decided if the bacteriophages are used. And there's not much data available from the Western countries, but I think Eastern countries have used bacteriophages for decades now, right? In the treatment, where we use antibiotics, they use bacteriophages. Do you think that is something that we could use in the Western countries in the future?
Sorry, could you hear me? I'm sorry.
It had something to do with phage?
If you can just like go.
I'm really sorry. So the topic...
No, it's my fault. I have trouble hearing.
Can you hear me now?
I was relying on you. I'm sorry.
Do you hear me?
Now we can hear you.
I can hear
louder.
you, but...
Okay, perfect. Just the question to bacteriophages. You said it would be an alternative for antibiotics that we can use these days. And it sounded like it's rarely used. And if I'm informed correctly, in Germany at least, we use it not as personalized medicine, but like individual patient medicine. So it has to be decided for each patient if the bacteriophages would be usable or used. And it's not very common. But I think Eastern countries have used bacteriophages for decades now. Where we used antibiotics, they mostly used bacteriophages. Do you think that is something for the future that we could use to tackle the problem, to still be able to treat patients so we don't have that many deaths related to antimicrobial resistance?
Well, I mean, you're right. It was... In fact, the earlier use of bacteriophage, it was in Eastern Europe where it was used extensively. More recently, there's been targeted use. I know I'm familiar with some of the work that's been done in the United States and a very active research approach to that, using it for very specific, highly resistant, very difficult to treat infections that might be fatal otherwise. Right now, at least in the United States, it's at a stage where it's very labor intensive, where they basically identify a bacteriophage that would work for a particular patient. And in that sense, in that setting, they've had some very dramatic and successful outcomes. For a general population, we're not there yet. I mean, there might be a way in the future that it should be explored to see whether it is one of the avenues that could be used in specific circumstance. But for treating infections broadly, I don't really see an application of it at this point. But I think the research on it, I think research should continue in multiple avenues because we don't know where there might be some breakthroughs.
Yeah, I have a question. I've been following throughout my adult life some of these questions, like the 5 years that we would have a hydrogen car. That's for 40 years. We've been promised that also that inflation is going to eat up all our savings. We've been promised that for 40 years and are currently being promised. And antimicrobial resistance is going to kill us all. That's also a threat that's been existing for the last 40 or 50 years, at least in the public space. So some of the obvious solutions, as in not using it in farming and so forth, are very obvious. My question is, do we also need a different business model so that it becomes more rewarding for the research and the commercial production? So instead of coupling the reimbursement to the amount of material sold, as you have shown, where basically the interest is to put out as much as possible, can we couple the reimbursement more to the quality of the solution, so that it actually becomes economically viable to produce a new antibiotic, even if it's rarely used?
Do the market incentives wrong? Should we be?
Right. Well, I agree that the incentives are not right. In fact, there's a bill right now before Congress, or that probably will be resubmitted with the Pasteur Act, to look at providing support to pharmaceutical companies or other groups for developing antimicrobials, but that would then be used in a limited way. And I'm not sure whether...
I think it was simply about trying to change market incentives to reward outcomes instead of just simply mass R&D. Is that correct? Yeah. Yeah.
Yes, I mean, I think we do need to look. We need to look at the outcomes from this.
So I'll ask another question, if I might. You know, the threat of antimicrobial resistance reminds me of a little bit of a great novel written by the American novelist Don DeLillo, and it was called White Noise, and there was a toxic event. It was a big threatening cloud, and it was moving towards a college town. And there's an amorphousness to the threat, and I guess I am interested to know what specific manifestation of resistance do you fear is going to break into our consciousness and perhaps change perception. So the one that you hear about in the press the most is drug-resistant TB. And I'm just curious if that looms particularly large in your view of the threats, or are there others that we're just not as aware of that perhaps we should be?
Well, I think drug-resistant tuberculosis has been and remains an issue. It really came into the fore, particularly around HIV, because HIV is such a potent cofactor in making people more vulnerable to tuberculosis. Drug-resistant tuberculosis is still a global problem. The issue about tuberculosis is it can also spread by the aerosol route. So if someone has drug-resistant tuberculosis, it can easily spread in a population. There currently is a lot of attention, but I think that TB is a more slowly spreading infection, and I think there's been more concern about some of the very acute and rapidly fatal infections in hospitalized patients. But drug-resistant tuberculosis will and does remain an issue. If, for example, the streptococcus or staphylococcus became resistant to all antibiotics, those certainly would cause immediate concern because they are so widespread, much more so than tuberculosis.
Questions? It's right over here.
I'm most interested in new substances and the research for new substances. We already tackled this problem. You mentioned it. Big pharmaceutical companies left the research years ago in the 90s, as far as I remember. Since the antimicrobial resistance is growing these days, do you see a change in strategy? Do pharmaceutical companies rediscover the research for new substances? Because at the end of the day, we need new substances. What else do we have to tackle multi-resistant strains? Isn't it? Do you see a change when you travel throughout the world and talk on conferences that pharmaceutical companies rediscover the research for new substances?
Are pharmaceuticals rediscovering research on new antimicrobials and so on and so forth because of this threat, from your experience?
On new antimicrobials? In general, were you talking about sepsis?
Everything.
Oh, yes. Right. I mean, most of the discovery has been by small groups. I think all of the major pharmaceutical companies have said there's no business model for making antimicrobials. So much of the discovery process, but it remains active. I mean, it's an area, the problem is the business model for then taking these molecules forward to do the very expensive testing that is required to show that they not only work, but may be as good as or better than. I mean, one of the problems is they've been required to show that they're better than the existing drugs and not just equivalent to them. And that has been a hurdle for many of them. But the business model is a problem. I mean, I think there's a lot of exciting science that's going on, but it requires science and the financial support to really move these forward.
Oliver, you're going to have to fix that business model problem. Okay. Okay. Last questions? We're very close to the end of our time. Very good.
Probably or hopefully this is a short question. First of all, thank you very much for this very clear presentation. I have the question, what about duration of use of antibiotics? Because in real life, doctors are prescribing antibiotics and many people don't take them the whole for 10 days, only for 1 or 2, and then they don't like it, or they are quite critical about that. Do you think that this is also a reason for increasing the antimicrobial resistance?
I think I understand what you're asking, whether when people are prescribed a course, they don't take the complete course. In fact, we don't know how long most antibiotics should be given. And in fact, it turns out if you look over time at the durations of recommended courses, they've generally shortened. Oftentimes, when duration is studied, it is found that a much shorter course is just as effective as a longer course. So people not taking, I mean, generally when people are prescribed a course of antibiotics, they are recommended to take the course that the doctor thinks will work. And in general, people should follow that. But not taking the entire course probably has not contributed to the development of antibiotic resistance. And in fact, many of the courses we take are probably too long, and much shorter courses are often. But it's very interesting. If you look at what's recommended, it's often 1 week, 10 days. They're often even numbers. And they weren't based on science when the initial recommendations were made. And I think that probably, what one shouldn't do is save, I mean, what you see in some countries where resources are really limited, people will buy some antibiotics. If they feel better after a day or two, they will stop because they don't want to use up this precious resource, and then they give it to a family member or someone else who develops some fever. That you don't want to do, to pass these antibiotics around, but shortening the course probably doesn't contribute to the overall development of resistance. Thank you.
Wait, can you wait for the mic?
I'll bring you.
Hi. So I know you had mentioned that a lot of healthy people have drug-resistant bacteria. I know healthcare workers, if you swab all their noses, most of them, or a very high percentage, would be MRSA positive. Are there recommendations, or is there a trade-off between treating healthy individuals that might be spreading this thing by having colonies of resistant bacteria versus even using more antibiotics?
Well, that gets into another whole area about decolonization if people are carrying multi-drug resistant infections. And I think in terms of treating healthy people, I don't think we're there in terms of wanting to give antibiotics in that setting. I think if a person were undergoing a procedure that was going to breach some of the barriers, one would want to take into account. For example, with prostate biopsies, there's been some studies looking at whether doing cultures in advance and basing the prophylactic antibiotics on the resistance profiles of the bacteria will reduce the number of cases of sepsis and death after the procedure. So if one's going to do an invasive procedure, yes, it may make sense. But I mean, it would be nice to get rid of it for the other people. But when you think about the cost of doing that, thus we have more precise ways of intervening, I think we're not there yet. But it's a good question to ask. Thank you.
Okay, if there are no more questions, I'm looking around the room. There appear to not be any more questions. I just want to thank you all for coming. For us, it has been great to come up here to vetting, in part because we get to meet an audience that we don't ordinarily see at the Academy. And we very much want to invite you to consider our other programming. It's kind of far away. Not all of it is in Wannsee. Some of it will be also in Mitte, maybe even more in vetting. But we do hope that you'll check our website to look for interesting events. That's americanacademy.de. And I just wanted to let you know that tomorrow our Holt Spring Fellow, Cristina Rivera Garza, a very distinguished American novelist, will give a talk entitled The Breath Archives, Effective Documents, Collective Memory, and the Afterlives of Femicide, an issue in which she has some very personal experience of. On Monday, May 8th, the distinguished nonfiction writer Ian Baruma will give a talk entitled The Collaborators, 3 Stories of Deception and Survival in Times of War. Both of those lectures start at 7:30, and if you'd like to sign up and attend, please do so. Otherwise, you can also watch them live over Zoom. Next week, we'll host a series of events with the renowned U.S. filmmaker Julie Taymor, including a special screening of one of her films at the Delphi Theater on the 14th. As you, I'm sure, all know, the Delphi Theater is enormous, and so you would be doing us an enormous favor if you could come and help us fill it up. We would really appreciate that. Anyway, again, please do look at our website. Thank you so much for coming today. This has been a real pleasure. Bye-bye.